which is by Regis, Joulia, who's a BHF-funded postdoctoral researcher in the lab of Professor Susan Norshard at the William Harvey, and he is going to talk to us on microvascular leakage disrupts localization of chemotactic cues and neutrophil diapedesis in vivo.
Okay, thank you. Is it working well? Yeah, you can see me.
Yeah.
Yeah, so thank you. Yes, so I'm Dr. Regis Joulia. I'm working at the Center for Microvascular Research at the William Harvey Research Institute. And today I'm going to speak about my project on the role of microvascular leakage and chemotactic cues and neutrophil diapedesis in vivo. So I don't have to remind this audience that during acute inflammation, the increased vascular leakage or microvascular permeability and neutrophil migration are hallmark of innate immunity. And central to the regulation of this phenomenon are the endothelial cell and the layer that they are forming, and notably the associated integrin and junctional molecule such as ICAM1, JAM family, and VCADRN family. Sorry, apparently I'm not in full screen. That's it. Yeah. So, so the integrin molecule. So as you, so these two phenomena are temporally associated, but they are supported by distinct molecular mechanisms, such as the VCADRN tight junction complex. And it has been shown by the team of Dietmar Weber, for example, that the VCADRN residue Y685 supports vascular permeability and Y731 supports neutrophil migration. In this context, we know that neutrophil migration, when they are migrating through the endothelial cell barrier, can induce themselves vascular leakage. But the impact on vascular permeability on neutrophil trafficking is unknown, and from the aim of this project. And to do this, we extended our confocal and intravital platform to analyze simultaneously neutrophil migration in the master muscles and vascular leakage in parallel. We've injected 75 kilodalton extra IV. You will see on this video, so this is a post-capillary venule of the cremaster muscle post-IR injury. In the reperfusion period, you can see the neutrophil here on the left side, but they are going to migrate through the blood vessels, and the dextran here injected IV. Very rapidly post-reperfusion, you can see the dextran is accumulating into the interstitial tissue, and it's slowly captured by perivascular cells, mainly macrophages. So we analyze in parallel the two responses in different inflammatory scenarios, L1 beta, LTB4, and IR injury, whilst these three reactions exhibit similar level of neutrophil extravasation, which exhibit different level of vascular leakage response, with LTB4 and IR injury showing vascular leakage, whilst L1 beta very minor or very low. Interestingly, we found that the reaction associated with vascular leakage, LTB4 and IR injury, were associated with aberrant mode of neutrophil migration, and mainly neutrophil reverse TEM. So neutrophil, instead of migrating from the luminal side to the abluminal side of the vessel through the entry pore, start to migrate in the subendothelial space, as you can see in the video. But instead of fully migrating through the subendothelial space, they retract the cell body back into the lumen, detached from the lumen, and after leaving the circulation. This phenomenon could occur to almost 15 to almost 25% of all neutrophil TEM. We then ask the question, but do vascular permeability agent could trigger neutrophil reverse TEM in vivo? And to analyze this question, we used this model of locally applied L1 beta for 2 hours and applied topically the histamine on top of the cremaster muscles. As you can see on this image and the different graphs, the application of histamine on top of an L1 beta reaction induced a very strong tissue dextran leakage in the interstitial tissue, but didn't impact the global neutrophil extravasation. But again, interestingly, we observed that the application of histamine led to almost 20% of neutrophil reversing back into the circulation, indicating that the induction of microvascular leakage promotes neutrophil reverse migration. We then asked what could be the associated mechanism, and we hypothesized that this disruption of the chemotactic gradient could be involved. As you know, during acute inflammation, not IL-1 beta, for example, chemokines are produced inside the interstitial tissue, such as CXCL1, one of the main chemoattractants for neutrophil. And we asked whether vascular leakage could impact the localization of the CXCL1. We first investigated by ELISA the tissue level of CXCL1 and observed that on top of an IL-1 beta reaction, the histamine didn't impact significantly the tissue level of CXCL1. But we observed that the plasma level of CXCL1 was increased following histamine superfusion, indicating that pro-permeability agent could account for an excess of systemic CXCL1. To confirm this hypothesis, we just injected during an IL-1 beta plus histamine antibody blocking BETP molecules, so to block the histamine-induced permeability, and you can observe that the mice treated with blocking antibody show reduced level of CXCL1 comparing to control animal. But it led to a general question that could an extravascular protein produced in the interstitial tissue cross the blood, cross the endothelial cell barrier during vascular permeability? And to do this, we analyzed the trafficking of a small molecular weight protein, the dextran, 10 kilodalton dextran, is similar size as the chemokine, and the protocol was as following. We applied IL-1 beta for 2 hours, then injected the 75 kilodalton extra IV to visualize vascular leakage coming from the blood, applied topically then the 10 kilodalton dextran inside the interstitial tissue for 10 minutes and replace the superfusate by either histamine or vehicle and record for the next 30 minutes. So you can see on this video again the signal of the 75 kilodalton dextra injected IV. So it's retained inside the circulation. And as soon as we start to superfuse histamine on the cremaster, you can visualize very strong vascular leakage inside the tissue. This dextran is then slowly captured by perivascular cells. On the right side of the video, you can observe the similar, so the same exactly postcapillary venules. And this time you will see the signal of the 10 kilodalton dextran. You can observe that as soon as we start to superfuse it, it fills rapidly the whole tissue. And you will observe very quickly that following histamine superfusion, the signal disappears very quickly. And where this dextran is going, we after collected the plasma of this animal and observed that the mice treated with histamine show higher fluorescent plasma comparing to the mice treated with vehicle control, indicating that the dextran is leaked inside the blood circulation. To further confirm this hypothesis, we established a collaboration with Dr. Kenton Arkell and Professor David Bates in Nottingham to analyze if mathematically it was likely that a protein could cross the endothelial cell barrier from the interstitial tissue. This probability is defined by the Piclet number, or PE, calculating using the advective flux, that is basically the hydraulic flux and hydraulic velocity coming from the blood inside the interstitial tissue, the length of the endothelial cell cleft, and the diffusion flux specific for each molecule. If the P is more than 1, the advective flux is dominant and the molecule is repulsed inside the tissue. If P is around 1, neither flux is dominant. And if P is less than 1, diffusion flux is dominant and the molecule can cross from the interstitial tissue into the blood. So to make a long story short about mathematical calculation, we defined that in a homeostatic situation, the P of the 10 kilodalton dextran is about 0.8, indicating that a low passage of the molecule is possible from the interstitial tissue. But the situation changed dramatically during vascular permeability. We know that there will be an increased hydraulic flux coming from the blood, but the weakening of endothelial cell junction lead to a more important reduction in the hydraulic velocity. And all in all, this more important reduction in velocity lead to a reduced advective flux and decrease to the Piclet number to about 0.3, indicating that it's highly likely that the small molecular weight protein could cross the endothelial cell barrier. We also analyzed the trafficking of a real chemokine in vivo. So to do this, we injected IL-1 beta in the cremaster muscle in combination with human CXCL8, applied histamine or vehicle control, and analyzed specifically the presence of human chemokine in the plasma level. We observed that the mice treated with histamine locally show a higher concentration of plasma human chemokine comparing to vehicle control, about 30%. And again, if we treat the mice with blocking anti-VTP to block the histamine-induced permeability, we observe that the level of the human CXCL8 was reduced compared to control antibody-treated animals. Finally, to go back to if vascular leakage or the excess of CXCL1 are responsible for the phenomenon of neutrophil reverse TEM. We perform confocal intravital microscopy in Vic-adherent mutant mice. So these Vic-adherent, as I mentioned in introduction, mutant mice show a defect in histamine-induced permeability and developed by the team of Weber. And when we perform IL-1 beta plus histamine reaction in this animal, we observe a reduction in frequency of neutrophil reverse TEM. In addition, if we block the excess of CXCL1 in the blood circulation by injecting IV a low dose of blocking antibody against anti-CXCL1, again, we reduce the frequency of neutrophil reverse TEM. Conclusion, I hope I convinced you that we discovered that during acute hyperpermeability inflammation, the increased vascular leakage leads to a disruption of the chemotactic gradient and an increased CXCL1 in the blood. This increased blood chemokine lead to aberrant mode of neutrophil migration, with notably some going back to the lumen of the vessel and going back inside the circulation. And as we have shown in this publication, these neutrophils are highly activated, migrate to other organs where they are causing damage. On this, I would like to thank people from the Center for Microvascular Research, my supervisor, Professor Susan Oshar, Charlotte Owen-Mood, and Matthew Benoit-Voisin, the rest of our team, of course, our collaborator both in Germany and also in Nottingham, Kenton and David Bates, and our funding bodies, and you for your attention. And I will take any questions.
Fantastic. Thank you very much again for yet another brilliant talk. Does anybody have any questions at all for Regis? No, perhaps I could just start then. So you said there right at the end that you think that the reverse transmigration might be to increase activated neutrophils in the bloodstream. So do you think that happens physiologically, and when might that happen?
Well, physiologically, yeah, physiologically for now, we didn't find any evidence of, we find always this activated neutrophil reversing back in pathological scenario. In physiological scenario, we could think that maybe these neutrophils are actually there to actually alarm the system that something is going on. So, but we didn't find so far any physiological reaction.
Okay, thank you. Does anybody else have any other questions? So Charlotte is asking what your imaging setup is.
My imaging setup, oh yeah, so we are using confocal, so this is normal confocal microscope with an upright objective that have been designed for intravital microscopy.
Okay, thank you. And Tim Miller is saying in vitro neutrophils will reverse following TNDF or INF stimulation. What other factors other than CXCL1 might be at work? Well,
we have shown in the past, for example, that the disruption of integrin molecules such as JAMC, for example, was crucial for the phenomenon of neutrophil reverse TEM. So for now, we have evidence that junctional molecule and chemokine are important. After other factors, for now, we're not sure.
Okay, thank you. And we just have one last question, and we'll leave it at that. So Nina Kalia has said, it looked like your 10 kilodalton dextran leaked out and disappeared from the interstitium straight away in the intravital video, and she's just questioning that is what she saw.
Yeah, it's disappearing very quickly. So so basically we think that basically it's leaking very quickly inside the blood lumen, and since basically the circulation is take it away very quickly. So that's why you see basically a disappearance of the dextran, because it's just in the circulation, it's washed away very quickly.
Fantastic, thank you very much. I think in the interest of time, we should leave it there. There are more questions on the chat, so perhaps Regis, you could get a chance to answer those later. So